Good day, everyone. Today, I'm going to discuss with you the Prelab discussion for the first part of the experiment 5, which is on the characterization of bacteria as a whole, or identification of bacteria. Okay, but for this first part, we will have the cultural, as well as the morphological characterization, as well as the staining techniques that you will expect to perform in the first part of experiment 5. So here I will be discussing the following. So first is on the background of the experiment, followed by the objectives and some reminders on the methodology. Okay, so in detection of an unknown bacteria, okay, so the first thing that you're going to do is to check their culture, okay, specifically the agar culture of this bacteria. So the bacteria is first classified according to certain growth patterns, okay, in different culture media. Okay, so for example, we can grow them in broth or in slant. Okay, so here are examples of these slants, okay, and their particular group. Okay, so here is the filiform growth, okay, which is a thread-like group, okay, thread-like root, so it looks like a thread, a thin thread, okay, or it can be arborescent or tree-like because of the roots, okay. Okay, so you can actually have a single line of streak here. Tapos you can observe the tree-like structures 18 to 24 hours after, and then they can be beaded, okay, or look like beads. Sorry, it's not clear in the photo, but they look like beads. Or it can be effuse or spreading. Okay, so there's an even spreading of the microbes coming from the single stem. Or it can be a rhizoid type of group. Okay, as you can see here, the observation for the rhizoids. Or it can be spiny like this. So they look like tiny spines on the line of inoculation. Okay, ayun. So in terms of the colonies naman on the plate, okay, you can actually characterize based on the size of the colony. Okay, so it can be small, it can be slight, it can be large, it can be moderately large. It's up to how you actually observe it. Okay, so ayun. So you can look at the um, multiple colonies, okay, for you to um, verify, okay, the size of the colony. Next is on the shape, which is shown in the figure above. So here we have the circular shape. We have here irregular shape. Okay, we have here filamentous shape or rhizoid shape. Okay, so these are the examples of the shapes of microbial colonies. Okay, so next is on the surface of the colonies. Okay, so here we can have an entire we can have a dull, a shiny, smooth, or contoured surface. Yeah, so you need to look closer to the microbial colony. And also using the following for the edge or the margin. So just the edges of the colonies, you can have it as entire. It can be wavy or undulate. It can be lobate. It can be curled or filamentous or filiform. Okay, so you need to look closer in order to observe the following columns in your unknown. Okay. So next is on the elevation. Okay, so how high is the growth in in the colony? So first is on the flat. So you can actually see this on the sideways. Is the side slants, gilded, okay, or it can be simple raised, or it can have an umbellate or having a knobby protuberance at the center, so it looks like a normal. Next, it's the other way around, so it's cateriform, so there's a dent at the middle. Or it can be a convex like this, or it can be a cushion-shaped pulmonate like this. Okay, so you need to look at the side. Okay, and then for the broth, you can actually look at it as based on consistency. Should it be a doughy consistency? Is it hard or is it sticky consistency? So you can actually have a quick feel of the microbial culture in broth. Okay. So next is on their capacity to transmit light, especially those in broth. So is it iridescent? Okay. So most of the light pass through the culture as pass-through. It can be translucent, so only a small fraction is passed through the culture. Or is it opaque, as in the light cannot pass through. Okay? So ayun, so that's according to the capacity to transmit. Okay? So examples we have here, enterofocus fascicium grown in nutrient agar. So we can actually see these colonies here. Okay, so as we can see, it's white. They are white, colored white. So they are circular. Okay, so they are convex based on site observation, and they are relatively smooth and have an intermargins, as in spherical shape. Okay, yeah. So they are found in human and animal feces. Okay, and so here we have other examples. So we have here Bacillus species. So in letter A, this is Bacillus cereus. This, as you can see, it is gray, it is granular, and the colonies are irregular. Okay, so as you can see here, the streak, okay, and then of course the growth patterns. Ayun. So letter B, we have here an Bacillus anthracis. Okay, so here they also resemble the cereus. Okay, however, they are much smaller. Okay? They are irregular edges. Okay, they have same consistency as Cereus. Okay, however, they are very much smaller in terms of the colonies. In letter C, this is Bacillus mycoides. Okay, as you can see, they are rapidly spreading and the colonies are rhizoid-like. Okay, hindi siya spaghetti or sotang. Okay, rhizoid colony. So that is what it looks like. And then this is letter D. So this is a non-bacillus. So it looks like a contaminant. So as we can see, the colony is wrinkled and irregular shape. Okay, but the margin is wavy. So this is the wavy margin or edge. Okay. So here is another. See, this is Rhizobium leguminosarium. Okay. So the colonies are convex. Okay. They are circular yet filamentous. So filamentous at the edges. Okay. So there are hair-like spines protruding the surface. Okay. So this is an ambonate colony. Okay. So as we can see here, there's this at the middle, the knobby surface at the middle. Okay. So the one at the right is getting in there, but not yet quite. Okay. So ayun. So next is on morphological characterization. So when we say morphological characterization, these are the cellular as well as extracellular structures that may or may not be found dun sa inyong bacteria. So here we have the mechanism of motility, if they are able to move or not. Next is endospore formation or endospore detection. Okay. Next are the cellular inclusions as well as the coloration. Okay. So the first thing that is going to be performed in morphological characterization is staining. Okay. So here we have different staining for the bacterial cell wall, the bacterial capsule, and the flatula. Okay. So for bacterial cell wall, we have the very famous gram staining. Okay, and to verify the results of gram staining, we have the Degrerson's method and acid-fast staining. So all will be performed in this experiment. And then for bacterial capsule, we have the negative staining using in the ink and Anthony's method. And for the presence of the flagella, we can perform the hanging drop test, the motility band, the flagellar staining, or the Schaffer-Fulton stain. Right? So here are some of the preparations lang before you perform the staining methods. Okay, so first is that you need to have a living or preserved samples. So the good news is that your microbial samples are 18 to 24-hour cultures, meaning they are still living. So your goal is to observe, identify, and check for the movement. Okay. So of course, the type of microscopy available will be compound light microscope. So you will be needing a microscope for this particular experiment. Okay. So the first thing you need to do is to prepare wet mounts or hanging drop mounts. So this in this particular method, so the cells are suspended in suitable fluid, such as water, a broth, or a saline solution. Okay, so the fluid maintains the viability and provides a medium for the movement of the cells. Okay, so it provides a true assessment of the size, the shape, and arrangement. Okay, so once the fluid is added, there's a slide. Okay, the next thing that you're going to do is to add a small amount of your microbes in the broth, and then mix it evenly. And then after that, you add a cover slip, and you know, the next thing is for you to observe them under the microscope. So the met mounts is consists of two drop or two of your culture that is placed in the slide. So nasabi ko siya kanina. So the advantage is that the wet mounts are quick and easy to prepare. Okay, and you can easily observe them under the slide. So it can damage large cells. It is susceptible to drying. That's why after preparation of the wet mounts, you need to observe it as quickly as possible. Okay, also, your hands, okay, your fingers, okay, can contaminate the wet mounts if you're not careful. So make sure your hands are not in contact with the wet mounts. Okay, so here's an illustration of the hanging trap mounts. Ayun. So here we have a slide, a glass slide with a certain depression. So when you say certain depression, so there's a dent at the middle of the slide. Okay, so the next thing that you're going to do is to prepare a sealant, okay, or a Vaseline right here. And then the next thing that you're going to do is to drop a specimen in the cover slip and slowly assemble them as you can see right here. So this overcomes the disadvantages of the wet mounts because first is that it has less contact with the air. Okay, and there's a small amount of space lang where the microbes can move. Okay, but you can actually see it. So next, after that, you'll have to carefully put them in the stage of the microscope and observe it. Okay, so yeah. So next, preparation techniques are the fixed smears. Ayan. So smears for the smear technique. Okay, so you need to spread a thin film made from the liquid suspension of the cells on the slide. Okay, and then after you have placed a small amount of this of the broth with the cells, allow the slide to air dry. Okay, allow it to air dry, and then you can actually perform staining techniques now then. Or the bad news about the usual smear preparation is that at the time that it is exposed to the air, it can be contaminated, diba? Ayan, so therefore we can use other preparation techniques, smear techniques to overcome that. So first is the heat fixing. So in heat fixing, we use heat or heat in order to fix the cells into the slide. So heat the slide gently after it has been air dried. Okay, and here are some important functions. It kills the cells. Okay, it secures the specimen to the slide. So therefore, if, after heat fixing, even though you move this slide multiple times, the cells will not slide from the slide. It preserves also cellular components in the natural state with minimal distortion. Okay. Aside from heat, you can also use alcohol and formalin, okay, for fixation. Okay. And then for the staining smears, ayon. So it provide you can stain the smears in order to provide on contrast. So it can make inconspicuous features to stand out. So previously unobservable features, it will now be observable after staining. So here, the dyes are used to color a particular part of the cell, okay, affixing themselves on the cell parts, okay, through a chemical reaction. So examples of dyes that are used for microbial staining are the basic dyes or acidic dyes. So basic dyes are cationic dyes. Okay, they have a net positive charge, okay, and they are attracted towards the acidic negatively charged components on bacterial cell wall. Ayan. So upon the interaction, of course, the dye will stay on this particular cell walls. So for acidic, anionic, or anionic stains naman, they have overall negative charge and they are repelled by the acidic negatively charged components on bacterial cell walls. So examples pala of these negatively charged components are lipopolysaccharides. Ayan. So upon the interaction of the acidic dyes doon sa bacterial cell wall. Okay, so it's not the cell that is stained, but rather it's the environment, okay, that is stained. Okay, so ayun. So there we have we have the two different types of staining. We have the positive staining and negative staining. So in positive staining, so the stain is actually found in the cells because the stain is attracted to the cell wall. It sticks to the cell and gives color to the cell. Okay. Negative staining, on the other hand, its negatively charged dye is repelled by the negatively charged bacterial cell wall. So instead that the cell is stained, it is the background that is stained. Ayan. So technically, the background will have a color. Okay, and you will see the cell in contrast. Okay. So here are the comparison of the positive and negative stains. Ayun, so in appearance of the cell, so here's the positive staining. Ayun, so we can actually look at the cells since they are the colored ones. And then in negative staining naman, okay, the cells are colorless and clear. So here the background is not stained in the positive staining, but in negative staining, it is stained. So examples of dyes for positive staining, we have the crystal violet, we have methylene blue, safranin, and malachite green as examples of stains for the positive staining. And for acidic dyes naman, we have negocene and India ink. So they stain the background. So examples of types of stains for positive, we have the simple staining. Next, the differential staining such as gram staining, acid-fast staining, and spore staining. Again, for positive staining. And then for the special stains, we have capsule, flagella, spore, granules, and nucleic acid. Positive staining. And then for negative staining naman, we can use these two types. So capsule staining and spore staining are some examples of negative staining. And so in simple stains, you only need a single dye. You just add it to the smear and that's it. You may need to wash it first before observing it under the microscope. So this is E. coli that is stained with crystal violet. Tapos here naman we have Corynebacterium that is stained with methylene blue. So as you can see, these cells are the colored ones. Okay, ayun. So the next major type of staining you will do is the differential staining. So you will use the different colored stains to clearly contrast the cell types or the cell parts. Okay, so this is under the complex staining technique. So we have the gram staining, acid-fast staining, endospore staining, and capsule staining process for the differential stains. So first up is the gram stain, which differentiates gram-positive bacteria to gram-negative bacteria. So it remains the universal technique used to um, differentiate bacteria. So it permits ready differentiation of major categories of cells according to their cell wall. So here the purple are the gram-positive cells. The pink or red naman are the gram-negative. Okay, so you will need the following for the gram staining. So crystal violet, grams iodine, ethanol, safranin as your materials for this. Okay, ayun. So here in the first step, you need to add the crystal violet. Okay, and then you need to flood your bacterial meal with the crystal violet. Okay, so ayun. So here, both the gram-positive and the gram-negative are colored purple or violet at this case. Ayan. So both of the cell walls are stained with crystal violet. That's why they're both violet or purple. The second step is the mordant, which keeps the crystal violet in the bacterial cell wall. So in this case, upon addition of the gram iodine, so still the gram-positive and the gram-negative are still purple. Ayan, so here the iodine keeps the crystal violet intact to the peptidoglycan. Ayan. The alcohol will remove the color or it is also known as a decolorizer. Ayun. So after addition of the alcohol, the gram positive will stay purple while the gram-negative will become colorless. Okay, so here the alcohol weakens the outer wall or the outer layer of the bacteria. Okay, and in negative cells, the outer wall is weakened and the cell loses the dye, making it colorless. But for gram-positive cells, since the peptidoglycan is thick, okay, the crystal violet will remain. And then the counter stain, okay, or the safranin will be added. So ayun, upon discoloration, the gram-positive will still be purple, while the gram-negative will be red or pink. Ayun. So technically, in gram-negative cells, ayun, so the safranin will be bound to the peptidoglycan layer and with color pink or red. But in gram-positive, since it is still previously colored purple, ayun, the purple coloration masks the saffron. That's why it's purple pa rin. So, ayun. So, now, you will have a differentiated gram-positive and a gram-negative. Okay. So, here's an alternative to the gram staining. So, here's the Gregerson method. So, you need to mix a loop full of the culture with 1 to 2 drops of 3% potassium hydroxide. And so in this case, you'll just have to mix them together, raise and lift, just like beating an egg. That's a scrambled egg, parang ganon. Okay, so if the mixture is watery, okay, so that's gram positive. But if it is slimy, so it's gram negative. So, yeah, you need to perform this also for this for the confirmation of the gram stain. Okay, so next is the acid-fast staining. Ayun, so there are certain types of bacteria that are acid-fast, and then there are some that are not. Okay, so here we have the red cells as acid-fast. The blue cells naman are the non-acid-fast. Okay, so here it differentiates between an acid-fast bacteria and a non-acid-fast bacteria. Okay, so here, this is especially useful specifically for Mycobacterium tuberculosis and Mycobacterium leprae. Yun, so one is acid fast, the other is non-acid fast. Okay, so ayun. So in acid fast staining, you will have to use the following. So first is for you to cover your smear with carbonfusion. Okay. Steam them over boiling water for 8 minutes. Okay. So add additional stain if the stain boils off because of the steam. Yeah. So after that, ayun, after 8 minutes, so cool the slide and add acid alcohol. So it's a mixture of hydrochloric acid and ethanol actually, equal parts. So for 15 to 20 seconds, just wash it, the smear. Ayun. And then stop the decoloration and rinse briefly with water. Ayun. So then you will add the methylene blue as the counter stain. Okay, so just flood it. Okay. And then rinse briefly with water to remove the excess methylene. And then finally, you will have to blot dry this and examine under oil immersion objective for you to actually see if the bacteria is acid fast. Okay. So ayan. So finally, we have the special strains. So these techniques are used to detect certain structures such as spores and capsules. So they are special because they pinpoint a particular characteristic, such as the presence of an endospore, presence of a capsule, etc. So for endospore staining, ayun, so it is used to distinguish endospores from vegetative cells. Ayun, so again, the endospores are produced by certain cells for survival, diba? So, yeah, so endospores here are colored red, while the normal vegetative cells are blue. So it detects the endospore forming members of Bacillus and Clostridium. Okay, ayun. So here, ayun, the most common method I use is the endospore stain, is the Schiffer-Fulton endospore stain, okay, wherein we use the malachite green as the primary stain and then the safranin as the counter stain. Okay, so here we have the greenish blue-green right there as the color of the endospores, while the cells, the living cells, are stained as safranin, the pink one. Next is on the capsule. So the capsule is a protective layer outside the bacteria surrounding the cells. So they can be visualized using negative staining using India ink or India ink. So it is used to identify pathogens such as Cryptococcus bacteria. So here's the India ink capsule stain for the Cryptococcus. Again, since this is a negative staining, so it's the background that is stained and the cells are colorless. Okay, so we can also use the Antonis method for bacterial capsule. So as an alternative, so here the bacterial smear is prepared without heat fixing. So just air dry it, then flood it with crystal violet and wash with 20% copper sulfate. So after air drying, okay, you observe it under OIO. If the cells are blue violet or colorless, so it is the bacterial capsule, while the dark blue are the cells and the vegetative cells. And then we also have the flagellar staining. So technically, it is used to stain the for you to actually see the flagella. The thing with the flagella kasi is that they are very thin, they are very slender, filamentous, and they're used for movement. And so therefore, the normal staining techniques cannot detect the flagella. Kasi nga, sa manipis. So here the flagella must be enlarged first in order for you to see it, and then a coating is deposited on the outside of the filament, then stained. So this is the flagellar stain of Proteus vulgaris. A basic stain was used to build up the flagella and make it visible under normal microscope. Okay, so aside from the staining, we can also use the hanging drop technique just like a while ago. So here you can actually see the movement of the cells. Ayun, so that is an indication of the presence of the flagella since flagella is used for movement. Okay. Or you can also use the motility band. Okay, so in motility band, okay, so you just have to have a agar dip. Okay, so here is an agar. Okay, and then stab a needle together with inoculate bacteria to this particular side. Okay, so when the bacteria is able to move, they will disperse out from the point of inoculation. Okay, so that's an indication also that the bacteria has a flagella. Okay, so here are some of the different terms used to name the to classify the bacteria based on flagella present. So it's Mototricus, so it only has a single flagella at the end. It's Amphitricus, ayun. So we have here flagella at both the poles. Amphitricus, there are multiple flagella but can only be found on a single pole like this. And Peritricus naman, the flagella is all over the surface of the bacteria. Multiple. Sobrang dami. Okay, so that basically ends the Prelab discussion for the cultural and morphological characterization of bacteria. So good luck for your experiment and enjoy. Thank you.